Assalamualaikum and hi, my name is Farah Nadzirah Binti Amin with student ID 2020988821 and my supervisor is Dr. Safiyyah Binti Shahimi. So my topic for Viva presentation today is soaking and accelerated fermentation for phytic acid reduction in oat Avena sativa. So this is the outline that I will be present today. As for the introduction, oats have long been recognized as a healthy and nutritious cereal with high levels of soluble fiber and minerals. Phosphorus, potassium, and magnesium were the three highest minerals in rolled oats. Nutritional profile of oats is well balanced. It has a good amount of carbohydrates and high quality protein with a balanced amino acid profile. Antioxidants such as vitamin E, phytic acid, phenolic compounds, and avenantramides are also abundant in oats. As for the phytic acid is a substance present in grains, legumes, nuts, and seeds, among other plant foods including oats. Phytic acid is the storage form of phosphorus, an essential mineral used in energy production and the development of structural elements like cell membranes. Phytic acid creates insoluble complex with polycation due to the reactive phosphate groups linked to the inositol ring, making them inaccessible for intestinal absorption and regarded as an anti-nutrient. The problem statement of this study is oats are a highly nutritious cereal consumed by various ages. However, oats contain phytic acid which will bind to important minerals in the digestive tract and inhibit them from being absorbed by the body. So the significance of study is the most effective wet processing methods between soaking and accelerated fermentation on reduction of phytic acid levels in oats can be identified. Next is reducing the mineral deficits that may occur in the future due to the presence of phytic acid. The objective of this study are to observe the effect of lactic acid bacteria (LAB) that present during soaking and accelerated fermentation on the level of phytic acid, and to compare the effect of soaking and accelerated fermentation on the level of phytic acid and magnesium in oats. Here is the summarization of the literature review. So minerals are inorganic elements that cannot be generated by the body and must be obtained through diet. So phytic acid is an anti-nutrient molecule found in oats. It chelates positively charged mineral cations in oats due to its high polyanionic nature. So it will reduce the absorbability of these minerals in humans. So figure 1 shows the structure of phytic acid and a phytic chelate at neutral pH. So as we can see here, when it is in its single form, it is called phytic acid. However, when it is bind to the minerals such as the calcium, magnesium, ferrum, and zinc, it is called phytate. Fermentation process occur during soaking. However, the difference between fermentation and accelerated fermentation is during accelerated fermentation, endogenous phytates was activated and accumulated. And as microorganisms grow, microbial phytates also accumulated. So phytase is a type of enzyme that breaks down phytic acid into myoinositol and phosphoric acid. LAB were found to have the highest phytase activity among microorganisms. Therefore, fermentation using active LAB is more effective because the organic acid produced by the microorganisms will reduce the pH close to the optimum pH required for phytase activity. Okay, so here is the methodology of the study. First is sample preparation, next is bacterial growth medium preparation, standard preparation, then we come to the analysis, which is the determination of phytic acid, magnesium, pH, and LAB count of the oat sample before soaking. So for analysis, which is the magnesium content, will be analyzed by using the acid digestion method. Phytic acid analysis will be analyzed by using Davis and Reid method. Then the pH of the soaked solution was determined by using the pH meter, and the quantification of LAB was done on MRS agar by using the spread plate method. Next, we will undergo the soaking method. So here is the figure of a soaking process, and the volumetric flask is the standard preparation for AAS analysis to analyze magnesium, and the test tube the figure of test tube is the preparation of standard phytic acid for UV analysis. After that is the isolation of LAB, where LAB will be isolated in the incubator for 48 hours at 37 degrees Celsius as shown in the figure. Next is the cultivation of LAB. So the LAB will be cultivated in the nutrient broth filled in universal bottle. So there were 3 universal bottles, which is negative control, positive control, and the cultured LAB. So the negative control was filled with the nutrient broth and sterilized distilled water. The positive control and the cultured LAB were filled with nutrient broth, and the LAB colony picked from the previous soaking procedure. Next is the accelerated fermentation. So the cultured LAB will be added in the accelerated fermentation procedure. So the difference between accelerated fermentation and soaking procedure is soaking only filled with 100 gram of oat sample and 400 ml of sterilized distilled water. However, in accelerated fermentation procedure, there will be 100 gram of oat sample, 380 ml of sterilized distilled water, added with 20 ml of cultured LAB. So, the addition of cultured LAB differs the accelerated fermentation from the soaking procedure. Next is the detection of phytic acid and magnesium after accelerated fermentation and soaking. And lastly is the statistical analysis, which in this study, 1-way ANOVA was used. So that's all for method. Moving on to the result and discussion. For pH analysis, the pH obtained show a significant difference at P less than 0.05 from before and after soaking, as well as after accelerated fermentation. There is a decreasing value of the pH started from 6.33 before soaking, followed by 5.84 after soaking, and 5.45 after accelerated fermentation. The pH changes give optimal condition for the activation of endogenous phytics and microbial phytics. LAB quickly dropped the pH to a point where competent species cannot grow. So a reduction in pH cell was result from a rise in lactic acid bacteria. So as accelerated fermentation have the lowest pH, meaning that it has the highest content of lactic acid bacteria. Next, for phytic acid level of oat samples, the phytic acid level obtained in oat samples also shows a significant difference at P less than 0.05 from before and after soaking and after accelerated fermentation. The regression line for the phytic acid standard has a perfect fit since the value of R was close to 1. So based on the result, the level of phytic acid decreases after soaking and after accelerated fermentation. The IP-6-myoinositol 1,2,3,4,5,6 hexakisphosphate, which is the hexa form of phytic acid, is converted by phytases into lower forms like IP5, IP4, IP3, IP2, IP1, and myoinositol. As for the analysis of magnesium, the magnesium level obtained in oat samples also shows a significant difference at P less than 0.05 from before and after soaking and after accelerated fermentation. So the R-square for the magnesium standard curve, which is 0.9984, also close to 1 and indicates a good R-square value. So the results showed an increasing amount of magnesium as the fermentation process occurred. As the fermentation process occurred, the amount of phytic acid was decreased. The mineral chelation by phytic acid also would be decreased, thus increasing the magnesium bioavailability in oat. For the LAB count in oat samples, 3 dilutions were used for the LAB plating before soaking of oats, which are 10 to the power of -1, 10 to the power of -3, and 10 to the power of -5. However, no colonies have formed for the 3 dilutions. This is because the phenolic acids found in oats have an antibacterial, antioxidant, anti-inflammatory, anti-allergic, and anti-carcinogenic properties. Next, the dilution of the soaking solution used for the oat after soaking and after accelerated fermentation were 10 to the power of -3, 10 to the power of -4, and 10 to the power of -5. However, for dilution of 10 to the power of -3, the results was too numerous to count, while the dilutions of 10 to the power of -5 shows the result of too few to count. So, the dilutions of 10 to the power of -4 was used. The colony forming unit, which is the CFU per mil, was calculated based on the stated formula, and the results were recorded in the table. As accelerated fermentation did increase the quantity of LAB in the fermentation process, this study has successfully proven that the amount of LAB in accelerated fermentation is more than soaking. This is the figures of colonies formed on the agar plate for before soaking of oat, after soaking of oat, after accelerated fermentation, negative control and positive control. And on the right side is the colony count for the oat after soaking, which is 75 colonies, and after accelerated fermentation, which is 101 colonies. In conclusion, the effect of soaking and accelerated fermentation on the level of phytic acid and magnesium in oats were determined. After soaking, the level of phytic acid in oats slightly decreased from 0.24 to 0.22 mg per gram, while the magnesium level in oats increases from 0.16 to 0.17 mg per gram. After accelerated fermentation, the level of phytic acid in oat decreased significantly compared to after soaking, from 0.22 to 0.12 mg per gram. The magnesium level in oats also increases significantly compared to soaking, from 0.17 to 0.24 mg per gram. Due to the presence of more LAB in accelerated fermentation, this technique has demonstrated to be more effective compared to soaking. As for the recommendation, in future research, it is recommended to carry out the analysis with different types of minerals such as phosphorus and potassium, which are among the highest mineral content in oat. It also recommended to continue the study on different samples with high concentration of phytic acid such as soybean, peas, and corn. So this is the list of references that I have used during my research. Here is the Gantt chart for the research project. That's all from me. Thank you.